Good morning, students. Today we are going to discuss lesson number 2, that is structure of chromosome, cell cycle and cell division. For the better understanding of this lesson, we have divided this into two parts. In first part, we will discuss structure of chromosome, and in second, we will discuss what is cell division and cell cycle. Today, what we are going to study or learn are these points. What are chromosomes? Who discover chromosome? What is the structure of chromosome? Structure of DNA? And what are genes? Before going ahead, I will request all of you to please take out your notebooks and note down whichever points you feel are important. Meanwhile, I'm going to tell you what are the important diagrams that you're supposed to practice. Before going ahead, I would like to revise or recap few things which we have discussed in grade 9. Number 1 is nucleus. Student, you know this is the most important cell organelle located at the center of the cell, and it controls all the cell activities of a cell. To study nucleus, we have learned what are chromatin fibers and what is nucleolus. As you can see in the picture, the thread-like structures appear in purple color. It's called as chromatin, and the nucleolus is located at the center of this nucleus. This part is most important as per as the structure of chromosome is considered. Correct? Moving ahead, what we are going to discuss in this lesson is this. Complete conversion of chromatin fiber into chromosome is the main idea behind today's discussion. Right? If you see here, this chromatin material, it is scattered inside the nucleus. But here they are perfectly packed and arranged in a particular shape-like structure. We called it as chromosome. We can use an analogy here that before going for a picnic or a vacation, we will pack our clothes and the essential things in a pack in systematic manner. Then why it is necessary that this chromatin material need to be converted into chromosome? Because this chromatin material contains genes which need to be passed from one generation to another generation. Right? So if I want to define chromatin, it is the complex of DNA containing genes and protein. Before going ahead, let's see what is the definition of chromosome. It is highly condensed coiled chromatin fibers. As you can see in this diagram, these noodle-like structures, although they are not noodles, this is called as chromatin. If you unwind this chromatin, you will see that this chromatin is made up of some silver pearl-like structures. We call them as histone proteins, and a yellow color thread-like structure surrounding these histones, we called it as DNA. So we will pack this chromatin material into chromosome. So we can simply say this chromatin materials are condensed form. Right? In 1982, Walter Fleming, he studied first the chromosomes in the larva of salamander. Now when he take out the cells of salamander and studied it under microscope, which was old type, although he was succeed to saw a minute thread-like structures which are appear in colored. The problem with the ordinary microscope is, student, we are not able to see the living cell until and unless we stain it. The meaning of the word stain is to give color to the organelles. So even Fleming, he was not aware about those thread-like structures. So when he used the color, all those thread get colored, and to that colored bodies, Walter Fleming called the word chromosomes. Chroma means colored and soma means body. Apart from this word chromosome, he also coined the term mitosis, because what he observed was thread. The meaning of the word mitos is thread. Since he was not able to explain about the structure of chromosome, how they are made up of, and what is their basic function, because of unavailability of the microscope instruments and more detailed information. Later on, the two scientists, Sutton and Boveri, they independently developed their studies to explain chromosome, and we call that theory as chromosomal theory of inheritance. This is not the part of our syllabus, but you can remember. Okay, we will go ahead. As you know that chromosomes are made up of chromatin, we have to first discuss what is chromatin. Chromatin is made up of two important substances: DNA, that is deoxyribonucleic acid, constituting 40% of its total volume, and proteins 60%. The protein which is present in chromatin is highly specialized, and we called it as histone protein. Okay. DNA, that is deoxyribonucleic acid, it winds around the core of 8 histone protein, forming nucleosome. Now if you see here in this diagram, these silver color structures, they are what? Histone proteins. And this is the yellow color thread is the one which is wrapped around it. It's called as DNA. And if these two structures together, we call them as nucleosome. This is the more detailed structure of nucleosome in front of you. As you know that we just discussed that there is a octamer. Octamer stand for 8 histones. Now these blue circular structures, they are the histone proteins 1, 2, 3, 4, and remaining 4, they are present just below this upper 4, right? So total they are 8 histone proteins. If anybody asks you the question, name those histone protein, then those are H2A, H2B, H3 and H4. All 4 multiplied by 2, it will become 8. There is no need to remember their sequence, in which sequence they are arranged. So once these 8 ball-like structures, they placed one above the other, the DNA, that this red color part, if you can see, this red color ribbon-like structure is called as DNA. The DNA will be wrapped around it, and once it will get wrapped, to pack these 8 histone protein along with DNA, we will have a specialized histone called as H1 histone. It will zip this complex, and then we call this entire complex as nucleosome. correct? But for nucleosome, you will not forget the names of these histone protein plus the number, that is octa stand for 8. Now since we discuss about nucleosome, let's discuss about DNA. Student, the shape of the DNA molecule was first studied by lady scientist Rosalind Franklin in 1953. However, the structure was finally worked out by some other scientist because it was not clear. As you can see, this is X-ray chromatography technique by which Rosalind Franklin tried to explain the structure of DNA. The other two scientists named James Watson and Francis Crick, right? They both were the student of Rosalind Franklin. Later on, they studied the structure of DNA more in detail and propose double helical structure of DNA. The photo which is in front of you right now is the actual model which was proposed by these two scientists. For that, they were awarded the Nobel Prize in 1962. Let's discuss this structure of DNA in detail. As per the Watson and Crick model of DNA, DNA is made up of two strands. In this diagram, I want you to follow this laser point. This blue color part is called as first strand of DNA, and this second blue color part is another strand of DNA. So total there are two strands, and they are spiral, that is spring-like structure. Okay. In between these two strands, you can see these lines, colorful lines. We call them as base pairs. You can correlate this with the steps of a ladder, and these two are the backbone of the ladder. Second point, although you can see that there are two strands, that two different blue color ribbon like structures, they run anti-parallel to each other. I will explain you what the meaning of the word anti-parallel in next slide. But remember, both the strands which are spiral, they are anti-parallel to each other. And this entire structure of DNA is a long chain of nucleotide, or simply you can call it as polymer of nucleotide. The way star molecule you have studied in grade 9 is made up of glucose unit. So glucose is the simplest unit. If you arrange the number of glucose units in a chain, it will form starch. Similarly, if we form the chain of nucleotide, it will form a polymer chain, that is called as DNA. We will go ahead. This is what the nucleotide look like. Okay, it is made up of deoxyribose sugar. In this diagram, this pentagonal shape is called as deoxyribose sugar. Okay, second, it is made up of phosphate group. This is called as phosphate group, and it contains nitrogen bases. Okay, how many nitrogen bases? Adenine, guanine, cytosine and thymine. Correct. Now total there are 4 nitrogen bases. The meaning of the word nitrogen, that they contain the main chemical component as nitrogen. Okay. Now out of these 4, adenine and guanine, they are called as purines. I want you to write this word purines. Adenine and guanine They are called as purines, P-U-R-I-N-E-S. P-U-R-I-N-E-S. While cytosine and thymine, they are called as pyrimidines. They are called as pyrimidines. P-Y-R-I-M-I-D-I-N-E-S. Okay. Now there was a scientist called as Chargaff, and according to Chargaff, adenine will always form pair with thymine and guanine will always form cytosine. And why it is so important? Let's see. Before going to discuss about nitrogen bases, I want you remember these two terms. One is nucleoside and another is nucleotide. What is nucleoside? When it contains deoxyribose sugar and a nitrogen base. We called it as nucleoside. And what is nucleotide? Deoxyribose sugar plus nitrogen base plus phosphate group. So basic difference between these two terms is presence or absence of phosphate group. Okay? So each single DNA strand is composed of a repeating unit of nucleotides, right? And these are called as what? Nucleotides, right? Now each nucleotide, as I just told you, is made up of sugar, phosphate group and nitrogen base. These nucleotides, they are connected by each other by a specialized bond called as hydrogen bond. Okay, and this is called as base pairing. The meaning of the word base pair, as I just told you, adenine, guanine, cytosine and thymine, these are the 4 nitrogen bases. Adenine always form pair with thymine and guanine always form pair with cytosine, or vice versa. The most important thing as per as our board exam point of view is this: adenine and thymine will connect by 2 hydrogen bonds, 1 and 2, while cytosine and guanine, they are bonded by 3 hydrogen bond, 1, 2 and 3. Now another important 2 bonds which are present in the structure of DNA, student, is this. Number one is hydrogen, second is glycosidic bond. This glycosidic bond will connect the phosphate group with its sugar. Correct? Phosphate is attached with the sugar by which bond? Glycosidic. Now I want to connect one nucleotide with another because I want to form a chain. So this long chain of nucleotide, that means suppose this is one nucleotide, this is another nucleotide, correct? And these 2 nucleotides are connected by a special bond called as phosphodiester bond. So what you supposed to remember is these 3 important bonds: hydrogen bond between 2 nitrogen bases, glycosidic bond between phosphate and sugar, and phosphodiester bond between 2 different nucleotides. This is okay. Now always remember, the arrangement is like this: nitrogen bases are arranged in inward direction, correct, which extends to join the complementary nitrogen base of another strand. Thus the 2 strands together make a ladder-like arrangement with this nitrogen bases, forming runges, or you can simply use the word rungs. Okay, we will not use the word ladder. We call this one important point as rungs. Okay, now the most important, this is another molecular or chemical view in front of you, that how adenine and thymine, they are forming the 2 hydrogen bond, guanine and cytosine, they are forming 3 hydrogen bond. Okay, we'll move ahead. Now student, whenever cell divide, the first and foremost important thing is the controlling center need to be divided. And if nucleus need to be divided, the first step that take place is the formation of a new DNA from the parent one. Formation of new DNA, that is forming another copy of same DNA, is called as replication. It's called as what? Replication, as you can see the first line. Okay. That is replication. And this replication of DNA take place in the interphase of a cell division. What is interphase? That we will discuss in the next part of this presentation. Okay. And as you know, as you just studied, that DNA is made up of double helix, 2 spiral strands. So from either of the end, it will open. From the either of the end, the DNA will open and it will start forming the new strands. Now there are 3 different types of replication, but we are most concerned about what happen in human. In case of human, the replication is called as semi-conservative. The meaning of the word semi-conservative is one strand is old and another strand is new. Just note down this 4th point in your notebook. And now, right? Now here I want to tell you the basic difference between conservative and semi-conservative. Right? Suppose this is the main DNA or the cell which want to divide. ये उसका DNA है, right? Now this cell is going to divide. So the foremost important thing is DNA also need to divide. So what will happen? These 2 strands, the blue one, this is one strand, okay, which is going to the right side, and second is which is going to our left side, correct? These 2 strands will separate. एक तो ऊपर से separate होंगे या नीचे से. Once they get separate, उनके exactly opposite new strand form होगा, correct? And when that new strand is formed, both the strands will get separate. So in conservative mode of replication, you can see there are 2 copies of the same DNA strand is formed, but correct? But again the older strands, they are rejoined, and the newer strands again they get separate and they form separate copy of DNA. This is called as conservative. Now let's go to the semi-conservative. This is what is your older strand, correct? And once these 2 strands get separated, उसके सामने new strand will be formed. So these 2 are these blue strands. Once they get separate, उसी के सामने the second yellow color new strand is formed. Even this was another old strand and this is the new strand. So when parent DNA form the 2 different daughter copies of its own DNA, having one old strand and one new strand, we called it as semi-conservative mode of replication. Okay. Now look at this GIF carefully. See what is happening in this diagram. This blue color DNA helix unwind, correct? And once it get unwind, there is these 2 blue color structures. We call them as enzymes. They 2 enzymes, they come in action and then they will start forming the new strand. Can you see this red color new strand? So if I see here, one is old strand and one is new strand. Correct? So since there are formation of two different strands, I will call this as semi-conservative mode of replication. And remember, this will go throughout the length of DNA. Amazing fact, students, we are having 2.2 meter long DNA present in a single cell. So this process will continue till the last part of the DNA. If you have not understood this point, you can call me anytime. Okay? We'll go ahead. As we have seen the structure of chromatin, structure of DNA, let's study the structure of chromosome. It consists of two identical spirally coiled sister chromatids. As you can see in this diagram. Before going ahead, I request you to please avoid these few labels. Kindly, the important label for this diagram is the centromere. Please remember this one. Now, what will happen? This part is called as chromatid. This part is formed by the coiling and supercoiling of the chromatin fibers. Once they get coiled around each other, they will form this chromatid. This is one chromatid, this is another chromatid, and both the chromatids, they are joined by a central spherical structure. We called it as centromere. We called it as what? Centromere. So centromere is the point of attachment of chromatids. If anybody asks you the definition, it is the point of attachment of chromatids. Normally, in each and every cell, the chromosomes appear like this. This is called as single chromosome. Okay? This is called as what? Single chromosome. And this is called as duplicated chromosome. This is called as duplicated chromosome. The meaning of the word duplicated, the one chromosome is converted into another copy after the end of replication of DNA, right? And these are formed by the condensation of chromatin, as I just told you. The most important part, what you remember right now, this centromere is not only joining these sister chromatids. See, these are called as sister chromatid. These two are sister chromatid. These two are also sister chromatid. So this centromere is not only joining these sister chromatids, but they are also the joining center of fibers. They are called as kinetochore fibers. They are called as kinetochore fibers during cell division. During cell division, these kinetochore fibers will attach to. These are the type of chromosome based on the location of centromere. Based on the location of centromere. Okay, this is again extra information, but good to know information. If you see here, centromere is exactly at the center of two chromatids, two sister chromatids. Correct? If the centromere is exactly at the center of two chromatids, we called it as metacentric chromosome. We called it as metacentric. Meta, exactly, center. Okay. Second type is submetacentric. What does this mean? Centromere is shifted to either of the side of chromatids. Not exactly at the center, it will be slightly shift from the center. That is called as submetacentric. Third is acrocentric. The meaning of the word acrocentric, centromere will be shifted almost away from the center of the chromosome. Can you see here? यहाँ पे वो exactly center में था. Now this centromere will either shift to this side or this side, forming unequal chromatids. The one chromatid will having a longer size and the another will be of a smaller size. This is called as acrocentric. And the last one is telocentric. Telocentric chromosome means there is only presence of single chromatid and the centromere. The another sister chromatid is totally absent. We called such chromosome as telocentric chromosome. Okay. This is most important point when it comes to the board exam, sketch and label chromosome. So please read the instruction whether the question talk about single chromosome or duplicated chromosome. If the single chromosome is asked, you will simply draw one centromere and two chromatids, and your label will be centromere and arm. Instead of chromatid you can write arm also, but when it comes to duplicated chromosome we will not use the label arm, we will use the term sister chromatids and centromere. Remember, duplicate chromosome will have 4 chromatids, 1, 2, 3, 4, while single chromosome will have only 2 chromatids, 1 and 2. This is the interesting point, student, of this lesson. What is gene? As per the definition that you are supposed to remember as it is the specific sequence of nucleotides on a chromosome that encodes the particular feature of the body. Now, if you see in this diagram, if this is the chromosome, we will uncoil this chromosome so as to form a long chromatin. Now from chromatin, if we remove the histone part, then we will get a single thread of DNA. And out of this single thread of DNA, this particular part is called a gene. Again, a fact, interesting fact, we are having somewhere around 23,000 genes present on each and every DNA of each and every cell. Right? So it's really amazing thing, and the same thing we are going to use for DNA fingerprinting. We will check which genes are present in parent as well as in the daughter or a son. Correct? This gene is also useful to identify the genetic diseases, and we can treat those genetic diseases based on gene therapy. Correct? That's it from my side. I request all of you to go through this video twice, thrice, read the lesson, as it is very small part of the lesson I have covered today, and solve the job sheet and attempt the test. Thank you so much. Good day.